That was a project that worked both with the target species, in other words, cattle, but also a model species, in this case, mice. So there is a mouse model of trypanosomiasis that informs some of our thinking on the disease, but doesn't tell the complete story. So going into the project, we had located regions in the genome, both in mice and in cattle, where there were genes that had an impact on on the progression of the disease in that species. The regions defined to contain those genes were large, and so trying to narrow it down, there are a couple of approaches. One is to go for further genetic studies, which is possible in mice, and in fact was taken a long way on the mouse side prior to the project starting, because you can breed further generations and reduce the interval interest to something smaller and more tractable. Much more difficult in cattle, given the generation time and the cost of breeding in cattle. So on the cattle side, but also we did something similar in mice, you're looking to bring in orthogonal data, so data that looks at the problem from a different perspective. And the data we brought in was gene expression data. So in fact, when we wrote the project, expression analysis using array technology wasn't completely new, but it was in its infancy as far as large animals were concerned, such as cattle. So we were looking at changes in the expression of hundreds, in fact thousands of genes in response to infection in both mice and cattle. And the hypothesis underlying this is that the genes that are showing differences in expression between animals that are susceptible to the disease and animals that can tolerate or resist the disease might point to genes that are in fact causal of those differences. So what we were looking to do was to bring together those changes in gene expression with the genetic map information to see if one could get a coincidence. So genes that are changing in expression which map to the region that have been shown by genetic studies to contain the causal genes.